So brain cells, both neurons and glia, are remarkable. And one of the most extraordinary aspects of neurons is that they have what we call excitable membranes, which means that they can change the permeability of the phospholipid bilayer, that is the membrane that surrounds the structure. They can change the permeability, which allows for ions to go back and forth, for current to flow, right? For charged particles to move from one location to another. And they can carry messages in the form of electric current, from one neuron, all the way along its length on this long axonal component, where it's going to reach ultimately the axon terminal, where there'll be a conversion process that we'll talk about, where the arrival of the electrical depolarization, the current here, is going to actually result in the release of a chemical neurotransmitter that can transfer that information, be reinterpreted at this neuron and converted back into another electrical message. Or, we'll see one of the wonderful things about, the fact that it's chemical communication at a synapse is that it could also block, or inhibit, the transmission. There's lots of things that can be done at that synapse because it's chemicals that are being released there for one neuron to communicate with the next. But in order for a neuron, right, to carry an electrical message, there have to be reasons why, charged particles, again, we're going to be talking about sodium, which is Na+, which is a very common dietary constituent, right? We need salt in our diets. We get plenty of it, but we need salt. We're going to be talking about potassium, another ion, which you get from bananas and kale and all sorts of good things. And then we're going to also be talking about chloride, which is a negatively charged ion that comes in with sodium. Sodium chloride is table salt, right? They separate out into the separate, charged ionic constituents in a watery environment like, your cells. And one of the things that's so important about, this discussion of resting potentials and action potentials is that in order for an electrical message to be sent, there has to be some reason, some forces that are going to be driving the movement of these charged dietary constituents, these charged ions like sodium and potassium, across the membrane, to form a current that can work its way, down an axon to the end. So neurons expend a lot of energy to establish what's referred to as the resting potential. And this is a term that is often very confusing because it actually derives from the history of investigation into resting potentials and action potentials, much of it conducted at Woods Hole, Massachusetts, where they had a squid off the coast, during the summer months that would allow, for like very close examination of axons, because the squid neurons were quite large. These are invertebrates. They're not as bound by a bony, kind of skeleton. They have very large cables that allowed for experimentation and examination and, determination of the actual mechanics and what's going on, with both resting potentials and action potentials. In Chile, off the Chilean coast, off the coast of South America, there's another squid, the Humboldt squid, the giant squid, which was available actually all year round. And so a lot of neurophysiologists, electrophysiologists, people studying, how neurons carry electrical messages, how they're set up to carry them, and then how during an action potential there's actual flow of electric current, went down to Chile near Valpo on the coast, and they examined, how this process works. And what they found was, in order for, ions to cross and pass, there have to be changes in the permeability of this membrane. And remember, we've got a phospholipid bilayer, which forms the membrane of all the cells in your body, including neurons and glia, and all the organelles within cells, like nuclei, like the nucleus and, other structures. Embedded in that phospholipid bilayer, which is largely impermeable, right, to the passage of charged particles, are protein machines. And these protein machines include channels, right? These are proteins that span the membrane from the outside to the inside and they're like holes. They got pores through them that are selected that let in certain ions, for example, like only sodium or only potassium, right? These channels we're going to see can be gated. They could be closed under certain circumstances, and then if you change the environment, you can ungate them. You can have them open. Other channels are just open all the time. They're called non-gated channels, right? Then there are also transporters. These are, protein machines that span the impermeable phospholipid bilayer, but, with the input of chemical energy from a chemical known as ATP, which is generated by cellular respiration, by breathing, burning sugar in the presence of oxygen in your cells. In these little organelles that are known as mitochondria, they spew out ATP, and the ATP can come in, and allow for, this transporter protein to grab something from one side of a membrane and, kinetically move it to the other side against some kind of prevailing force. And this brings up that there are forces that are going to be driving charged particles from one location to another that are operational in this kind of, environment, bodily environment, right? Brain environment. And these include what's known as the concentration gradient force. That's simply stated, things that are in high concentration tend to move to areas of lower concentration. And so we're going to see that a neuron at rest, before it sends an electric message, is actually using a lot of energy to make for differences in concentration across the membrane, the extracellular outside the neuron, versus the intracellular inside the neuron, because those differences in concentration will drive the movement of these, charged particles, these ions, depending on, what those concentration gradients happen to be. Things will move, just by the force that exists from areas of high concentration to areas of low concentration. To give you a little insight into what's going on here, if you take like lemonade mix and you put it into a pitcher of water, well, you got high concentration right there. Even without stirring that, over time, all the little random motions of the chemicals that make up the lemonade mix would drive this sort of distribution, this sort of like more, consistent distribution throughout the pitcher. If it stayed in high concentration, they made little decisions to keep sticking around in that high concentration, we'd need new physics, right? So there's also something called the electrical force, which simply stated is that oppositely charged substances like sodium and chloride, right? Sodium is positively charged, chloride is negatively charged. They attract each other, right? While like charges, things like lots and lots of sodium together, lots of positive ions all together, repel each other, right? So we're going to see that when you set up in a neuron at rest, high concentrations on one side of a membrane, of a very highly positively charged substance and low concentrations on the inside, well, not only is the concentration gradient force going to drive, the substance, let's say sodium, from high concentration extracellularly to lower concentration inside, but also the fact that there's going to be a charge difference because these, substances, these ions that are distributed, in terms of concentration, carry charges, right? So opposite charges, remember, attract, and like charges repel each other. And finally, neurons, again, like I suggested, expend a great deal of energy to maintain both these concentration and charge differences across the impermeable phospholipid bilayer membrane. And there's a particular protein machine, a biological force, that is extremely important in sort of maintaining these concentration and charge differences across a neuron's membrane at rest. And this is a machine called, historically it's been called the sodium-potassium pump. And what it does is it grabs onto, let's say a sodium on the inside. And when, a chemical grabs onto another chemical, there's a shift in shape, right? And then there's let's say on the outside, on the extracellular side, a piece of it will grab onto a potassium ion, right? And then it'll shift shape, and then it'll get another sodium ion on the inside, and then another potassium ion, and then another sodium. So it'll get 2 potassium ions attached extracellularly, and then 3 sodium ions attached intracellularly, and when it does, it shifts to allow ATP, that energy molecule, to come in and to break a bond, in ATP. One of the phosphates is, basically that bond is broken, and that chemical energy is transferred to the kinetic movement. The 3 sodium ions are shoved out extracellularly, taken out of the cell, while the 2 potassium ions are actually brought inside. And what this does is it contributes to the concentration gradients and the electrical gradients across the membrane that determines the resting potential. And, that term, it's confusing, but like in Chile, for example, they would take one electrode, right? They called it a potential, and they'd stick it on the inside of the membrane, the intracellular side, and they'd take another electrode, a potential, and stick it on the outside, and they saw that there was a difference in charge because current would flow. It was more negative on the inside and more positive on the outside. So there's this charge difference, like a potential difference in charge, difference between the inside potential and the outside potential. And at the resting, before a neuron fired, that charge difference rested. It stayed the same. It was around 65 to 70 millivolts, thousandths of a volt more negative on the inside than it was on the outside. And think about that pump, right? That pump is kicking out 3 sodium ions for every 2 potassium ions that it brings in. Well, that's an unequal distribution of charges, right? You're kicking more positive ions out, utilizing ATP, than you're bringing in. That contributes to that difference in charge. Another thing that contributes to the difference in charge is the fact that inside a neuron, inside a cell, there are lots and lots of proteins, and fewer, that are found extracellularly. And proteins, because they're manufactured inside cells, they build all the machinery, including the pump and including all these other channels and everything else that are involved here. They're going to have a net negative charge because proteins are built of smaller units that are called amino acids. There are 20 kind of dietary essential amino acids, or 20 essential amino acids, actually. Some of them you can construct. And those, the majority of them have a negative charge. So proteins tend to have more of a negative charge attached to them because they're made of more negative pieces than positive pieces. And so they call these anions. They're negatively charged things. The proteins that are inside the cell, that also gives that interior more of a negative, cast sort of to begin with. And in a neuron at rest, right, where you have one potential outside and one potential inside, you see current flowing and it's staying kind of the same, and it's 70 millivolts more negative inside than it is outside, you're going to find a very high concentration of sodium on the outside because there are only a few holes or pores or non-gated channels in a neuron at rest that allow for sodium ions to move in. And there's a few more non-gated potassium channels, which allows, we're going to see potassium to sort of move out, actually. And the pump, of course, is operating all the time to kick 3 sodium out for every 2 potassium that it brings in. So there's this resting potential really refers to the fact that there is a difference in charge between the potential on the inside and the potential on the outside, the electrode in and the electrode out. The inside of the neuron is more negatively charged versus the outside, right around the membrane, by about 65 to 70 millivolts. And that's, a very energetic, ATP requiring, sort of circumstance that's necessary to set the neuron up, for ultimate changes in permeability in that membrane that will allow for action between the potentials, that will allow for currents to flow, and work their way down, from, the beginning of an axon, to the axon terminals.